the key thing of this degeneration, the Alzheimer's disease, is what we call the cognitive impairment. Or in a more plain language, it's a deficit in learning and memory. So we put together experiments, includes almost 100 tau mice from those 3 lines, and also we included C57 black 6, or we just call it black 6.
Yes,
just do that as a control. So in total, around 100 mice for an experiment which lasted for 9 months.
9 months?
Yes.
Wow.
This is important because, you know, for human, the neurodegenerative disease, the Alzheimer's disease, FTD, whatever things, it does not happen in younger people. God bless it. It does not happen to young people. It happens to senile population. So for studying this kind of thing, we cannot accelerate. We cannot skip this process. We have to wait. We have conducted hundreds of runs of behavior tests and all that other things.
Wow.
Let me just explain our experiments. We set 3 time points, at 3 months old, at 6 months old, and at 9 months old. So we focused on cognitive aspects, and we also focused on other general vital aspects of the mouse, such as the motor function, the anxiety, and other general behaviors.
Yes.
To put it in a more academic way, we set up several behavior tests, which includes the novel object recognition test for learning and memory, open field for general behavior, and the rotor road for motor functions. Okay.
So besides the behavior studies.
After that, after 9 months of tedious work, hard work, we conducted pathological studies.
Yes.
Using both staining and Western blot to see how the proteins expressed in the brain.
Okay.
But that's something we are going to talk about a little bit later. So at the very beginning, we conducted the open field test and also the rotor road to see the general behavior in the mouse. As expected, no alterations. For this no alterations, I have to explain that means the entire HUGO series, the 3 HUGO line, they do not deviate from the black 6. And also, within the 3 HUGO line, the mutant lines, they do not deviate from the wild type line. That means at 3 months of old, it's safely for us to say there is no behavioral pathology. The next time point is 6 months old.
6 months.
Now things are getting interesting. We didn't see too much of the motor dysfunction in those lines, neither compared to their own HUGO wild type or the black 6. But we did see some deficit in cognitive function, especially in the P301S mutation line.
Okay.
In the novel object recognition, we see that the H-tau P301S mutant mice, they didn't recognize the novel object in compare to the old object they have been playing with for, I think, 24 hours. Well, you know, the mouse, they like exploring new things. So if it can recognize new things, they will play with that for a longer time. But they didn't happen with the 301S mouse. That means they didn't remember what they have seen.
Cognitive deficit.
That is interesting. That is even more interesting. We didn't see anything in the P301L line at 6 months old.
Okay, maybe it's too early for it?
Yes, it's too early. And this is exactly happened in human patients.
Okay.
The human patients of FTD, the human patients with AD who bears the P301S mutation,
has an early onset.
Yes, has early onset and accelerated pathophysiology.
Okay.
That means at least in terms of the novel object recognition test, in terms of this episodic memory test, our HUGO mouse behave exactly like human. By the end of our experiments, 9 months old time point, we still didn't see any motor dysfunction in all those lines, but we did see the cognitive deficit in both P31L and P31S. Now it's catching up. The P31L is catching up. Eventually, the cognitive deficit happened as expected, yet slower than P31S. Also exactly like in human.
What about the wild type tau?
The wild type staying functional, clever, capable of recognizing the new things. By comparing the HUGO-Tau wild type to black 6 mouse, we didn't see any alterations. Not in cognition, not in general behavior, not in motor function. This is just the behavior side of the H-Tau series. By completion of the behavior set, thank you again, animal department. By completing the animal behavior test, we conducted pathological test using the brain tissue from all those 4 strains, wild type, HUGO wild type, P31L, and P31S. While you know, in human patients, the most important pathological hallmark by staining is what we called neurofibrillary tangle, which is a tau aggregation inside the neurons.
Yes.
So this time we expect to see the same thing in those mouse models. For this experiment, we designed 2 arms of the experiment. One arm, of course, the staining is the major part.
Yes.
Using 2 types of antibodies. One is the HT7, which is one of the gold standard for tau detection.
Yes.
It detects only human tau, not mouse tau. Another is 88, detects only hyperphosphorylated tau, which is the pathological related tau, and it's majorly human. For another arm of our experiment, we designed Western blotting, which is mainly for differentiating the wild type mouse and the humanized mouse to see, well, at the genetic level, we validated that the human tau expressed only in the HUGO and the mouse tau expressed only in mouse, but we still want to see at protein level if it is really the thing. So for this purpose, we use another antibody we call tau5 as a control. Tau5 detects both mutant tau and human tau.
Okay.
So with this sound complicated design of study, let's see the result. This is the staining result. And as expected, using the HT7 antibody, we detected nothing in the black 6 mouse.
Of course.
Of course, it's expected there is no human tau. And also as expected, we detected a bunch of tau in the H-tau wild type. That means the human tau is expressed as expected.
Yes.
And interestingly, we detected also HT7 signal in the P31L and P31S mutant lines.
Both mutant.
Yes, but the interesting thing is the pattern of tau expression is different. Here you can see many of these tau aggregations in the neurons.
Yes.
By the way, I didn't explain, this is the hippocampus of the mouse.
Okay.
Hippocampus, the central of memory.
Yes.
Is our behavior test related. We see this aggregation in the P31L and the P31S, indicating there is a tau aggregation happening inside those neurons. This is part of the validation. With this HT7 antibodies, we conducted our Western blotting, and I think this result is self-explanatory. Yes, you see the HT7, there was nothing in the black 6. However, there is a bunch of things in our HUGO series. And by using tau5, we also see in all of these strains, the tau was detected. However, interestingly, in different patterns. That means the tau isoforms, the tau bands in the HUGO series differentiate from the black 6. Also, another evidence corroborating that we have human tau inside those strains.
Yes.
Not just the mutant tau.
Yes, and all the human isoforms.
Yes, and all the human isoforms, which is such a beautiful thing in those HUGO strains. And after the HT7 detection, of course, we ought to detect the pathological tau. We ought to use the 88 to detect it. And as expected, we didn't see much in the black 6.
As expected.
As expected. And this time, we didn't see much also in the human tau wild type. As expected, there is no pathology. They behave normally, just like the black 6.
Yeah.
And we detected a bunch of signal in the P31L and the P31S. Since 88 is specially detecting the phosphorylated tau, we could safely say that there are a severe hyperphosphorylation process of tau happening in the brains, especially in the hippocampus of those mouse.
Which is characteristic of the AD.
Of the AD, of the FTD, of any tau-related pathology. We gathered tons of data from those mouse, and we now have very solid data saying that the HUGO series, the HUGO mouse models successfully simulate human tau-related disease. For the last several years, as we understand that the majority of AD therapeutic trials focused on A beta side. But that does not mean we forget about tau.
Yeah.
Cyagen help you discover life.